Have you ever wondered what happens when we combine the building blocks of life with the materials of modern technology? It's a field buzzing with incredible possibilities. Scientists and engineers are on a fascinating quest to merge the soft, living world of biology with the hard, structured world of materials science. Imagine creating a computer chip that can seamlessly interact with your body, or a medical implant that your cells recognize as a friend, not a foe. This is not science fiction. It is the frontier of nanoscience. Achieving this dream requires us to understand how to arrange biological molecules such as proteins and peptides onto solid surfaces with incredible precision, creating a perfect bridge between these two very different realms. The challenge is a big one. Living things are dynamic, flexible, and often thrive in water. In contrast, the materials we use for electronics and devices, like graphite or silicon, are rigid, flat, and unyielding. How can we convince delicate biological molecules to organize themselves neatly on these foreign surfaces? It's like trying to get a group of playful puppies to line up perfectly on a smooth, slippery floor. Scientists have long been exploring this puzzle, knowing that cracking the code would unlock a new generation of smart materials. The goal is to create bio-hybrid systems where biological function and material properties work together in harmony, opening doors to advanced biosensors, new medical tools, and even more efficient energy technologies. To achieve this, researchers are turning to some of the smallest and most fundamental components of life, peptides. These are short chains of amino acids, the very same building blocks that make up the much larger proteins in our bodies. Think of them as tiny biological Legos. Because they are smaller and simpler than full-size proteins, peptides are easier to design, create, and study. Scientists can carefully select which amino acid bricks to use and in what order, giving them precise control over the peptide's properties. By understanding how these custom-designed peptides behave, we can learn the fundamental rules that govern how life builds its intricate structures, and then use those rules for our own technological creations. The journey to control these tiny molecules on a flat surface is a delicate dance of chemistry and physics. It involves understanding forces at a scale 1,000 times smaller than the width of a human hair. Everything matters: the type of amino acids used, the exact atomic structure of the surface material, and, as a recent breakthrough has shown, the environment in which this assembly happens. Scientists are discovering that the secret ingredient, the invisible choreographer guiding this intricate dance, might be something we see every day: water. It turns out that this simple molecule plays a surprisingly complex and absolutely essential role in helping life's building blocks find their place in the world of technology. So, what exactly are these magical peptides? As we mentioned, they are short chains of amino acids. In a recent groundbreaking study published on June 26, 2025, a team of researchers focused on creating very specific peptides using just two types of amino acid building blocks, tyrosine and histidine. Each of these amino acids has a unique personality. Tyrosine is what scientists call hydrophobic, which is a fancy way of saying it's water repelling. It tries to avoid water whenever possible, much like oil does. On the other hand, histidine is hydrophilic, meaning it's water attracting. It loves to be surrounded by water molecules. This contrast between water-loving and water-fearing parts is the key to their special behavior. The researchers from Kanazawa University and their collaborators were like molecular architects. They didn't just mix these amino acids randomly, they designed peptides with a repeating, alternating pattern of tyrosine, which we call Y, and histidine, which we call H. They created chains like YH, 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 and even longer versions. By changing the order and the number of these repeating units, they could fine-tune how the peptides would act when placed on a surface. This precise design is what makes modern nanoscience so powerful. It allows scientists to create bespoke molecules that are programmed to perform a specific task, in this case, to line up perfectly on a flat material like graphite, which is the same stuff found in your pencil lead. Why is this arrangement so important? When these peptides are placed onto a graphite surface, their personalities come into play. The water-fearing tyrosine parts are naturally drawn to the flat, water-repelling graphite, wanting to lie down and stick to it. Meanwhile, the water-loving histidine parts prefer to face outward, interacting with the surrounding water. This push and pull dynamic within each tiny peptide chain is the first step in creating order. It's a self-organizing principle guided by the fundamental properties of the molecules themselves. The scientists aren't pushing them into place one by one. They are creating the perfect conditions for the peptides to spontaneously build the structure all on their own. This process is a beautiful example of self-assembly, a core concept in nanotechnology. It's nature's way of building complex structures, from a simple snowflake to a living cell, without a tiny construction crew. By harnessing this principle, scientists can create incredibly detailed patterns at the nanoscale. The peptides, with their alternating hydrophobic and hydrophilic segments, are designed to not only stick to the surface, but also to connect with each other. They form orderly, straight lines that follow the underlying atomic grid of the graphite, creating a perfectly aligned nanoscaffolding. But as the researchers discovered, the peptides couldn't achieve this remarkable feat alone. They needed help from their environment. This is where water takes center stage, revealing its secret role as the master organizer in this nanoscale world. It's easy to think of water as just a passive liquid, the background in which chemistry happens. But at this tiny scale, water is an active and essential participant. When the peptides begin to assemble on the graphite surface, the water molecules don't just stand by and watch. Instead, they form a highly structured, dynamic layer around the peptides, known as a hydration shell. This isn't just a random coating of water, it's an organized molecular blanket that wraps around each peptide assembly, influencing every step of the process, and stabilizing the final, beautiful structure. The hydration shell is incredibly clever. It's not uniform, it's heterogeneous, meaning it's different in different places. The water molecules arrange themselves precisely around the water-loving histidine parts of the peptides, forming strong hydrogen bonds and creating what scientists call binding pockets. These pockets are like tiny, custom-made gloves that hold the peptides in place and mediate their interactions with each other. At the same time, the water helps push the water-fearing tyrosine parts toward the graphite. This cooperative action ensures that the peptides lie flat, fully extended, and perfectly aligned with their neighbors, creating those long, crystalline lines that scientists were hoping to see. Furthermore, water provides the necessary flexibility for the peptides to get into position. Imagine trying to arrange dry spaghetti noodles into a pattern. They are brittle and would likely snap. But if you soak them in water, they become flexible and can be easily arranged. Water does something similar for the peptides, giving them conformational flexibility. It lubricates their movements, allowing them to wiggle and adjust themselves during the assembly process until they find the most stable, low-energy position. Without this watery cushion, the peptides might get stuck in the wrong arrangement or clump together randomly. Water is the gentle guide that allows order to emerge from molecular chaos. To truly understand this, scientists combined powerful experiments with sophisticated computer simulations. Using a technique called frequency-modulated atomic force microscopy, or FMAFM, they could actually see these individual peptide lines on the surface. More advanced three-dimensional AFM methods allowed them to visualize the structured water layers enveloping the peptides. The computer models, known as molecular dynamic simulations, confirmed what the microscope saw, showing how the interplay between the peptides, the graphite surface, and the intricate hydrogen bond network of the water layer was the key to creating and stabilizing these remarkable nanoarchitectures. It was clear, water was not just the solvent, it was the architect. The ability to control molecules at this level of precision opens up a universe of exciting future applications. Now that we understand how to use water to help peptides form perfect lines, we can start using these lines as a template, like a nanoscale ruler or scaffold. Imagine using these ordered peptide lattices to organize other nanoparticles, such as tiny bits of metal or quantum dots, with sub-nanometer accuracy. Arranging quantum dots in such a precise way could allow scientists to study and control quantum effects, potentially leading to new forms of computation or ultra-sensitive sensors. It's like having a molecular assembly line for building the technologies of tomorrow. This discovery has huge potential in the medical field as well. By designing peptides that can form these ordered structures on the surfaces of medical implants, we could create materials that are more compatible with the human body. These peptide-coated surfaces could guide how cells attach and grow, promoting better healing and integration of the device. Furthermore, by incorporating specific amino acids, these structures could be designed to act like enzymes, the natural catalysts in our bodies. This could lead to new types of biosensors that can detect specific molecules in a blood sample with incredible accuracy, or even create smart surfaces that can trigger a chemical reaction on demand. The tools that made this discovery possible are just as amazing as the discovery itself. Atomic force microscopy allows us to see and even touch things at the atomic scale, giving us an unprecedented window into this invisible world. When combined with the predictive power of computer simulations, scientists can now design, build, and verify these nanoscale structures with confidence. This synergy between experimental observation and theoretical modeling is accelerating the pace of innovation, allowing us to unravel nature's most complex secrets and apply them to solve real-world problems in medicine, electronics, and energy. It's a testament to how far science has come. Ultimately, this breakthrough is a beautiful story of collaboration, not just between scientists from different countries and disciplines, but between science and nature itself. By carefully observing and learning from the subtle dance between peptides and water, we are learning to speak nature's language of self-assembly. Understanding water's critical role is a major step forward, reminding us that sometimes the most profound secrets are hidden in the most common substances around us. As we continue to explore how different molecular ingredients influence this process, we move closer to a future where we can build a better world, one molecule at a time.